Okay, we're just waiting for people to come in from the waiting room on our online webinar today. Thank you for coming, everyone who's in the room. Okay, we'll go ahead and get started slowly as people continue to come in from the webinar waiting room. Welcome to our Thursday scientific seminar series at Carey Institute. And this is a hybrid presentation. And so we have people in the room and people joining us online. We will hold questions until the end. And you may put your questions in the chat or question and answer part online. And we will field those at the end of the talk. And if you have any logistical issues online, please put that in the chat, as well as if you would like to tell us where you're joining us from. All right, so I am pleased to introduce this week Dr. Sarah Diamond. She's an associate professor at Case Western Reserve University in Cleveland, Ohio, and also an ESA, Ecological Society of America Early Career Fellow as of 2018. Right? Dr. Diamond's an evolutionary ecologist with a strong entomological focus, which we'll hear more about. And I'll say I've been following her work over the past few years. In particular, she's published a series of papers that have helped redefine our understanding of contemporary evolution, how fast things actually might be evolving, and really highlighted how urbanization is both a driver of that and can be leveraged as a tool to really better understand it and to study mechanisms of population and community persistence. I'm really excited to hear the talk today, and I'll let her further describe what she's been up to. So welcome.
Thank you so much for that lovely introduction, and thanks to all of you, both in person and online folks, for coming to the talk today. So I am really excited to share kind of an overview and synthesis of some of the work that we've been doing, looking at how organisms respond to anthropogenic change, in particular thinking about how they're responding to urban heat island effects. And so we think about organisms being relatively well adapted to their environment. And I've just kind of picked a smattering of some of my favorite examples of extreme adaptations here. But the environment changes, and this poses challenges to these tight associations between phenotype and environment. And this ranges everything from sort of catastrophic events such as asteroid collisions, or perhaps more subtle changes such as the little ice age depicted over here. Now, the environment has always changed, but it's changing at an unprecedented rate and by an unprecedented magnitude over contemporary timescales. So you imagine the sort of extreme pressures that this is putting on populations, the potential to disrupt that environment phenotype relationship. And so a big open question is how populations are able to cope with these changes in the environment, particularly sort of contemporary global changes that are impinging on populations. And so if we just take a simple toy example here, where we have a depiction of a relatively well-adapted population in its ancestral environment. So we have fitness depicted as a function of trait values. We have a distribution here, and we have a population basically sitting right on its fitness peak, so a well-adapted population. With environmental change and the generation of these novel environments, we can shift that distribution and the potential location of that fitness optimum. In this case, the environment is now just kind of overall degraded, and we see a drop in the maximum fitness that population can achieve. And there's not really much to be done about this except for restoration of that particular environment. Now, as an alternative scenario, we can have the case where the distribution of the novel environment shifts. And so there's the potential to still achieve relatively high maximal fitness in a comparable fashion to the ancestral environment. It's just the location has shifted along the trait value axis. And so because we have this sort of ancestrally adapted population with a particular trait value, with the shifting to this novel environment, fitness is going to drop. And so the key question is kind of whether and how organisms can enact these compensatory mechanisms to allow them to get back up to this fitness peak in the novel environment. And one way they can do that is through phenotypic plasticity. And here we're talking about within generation changes in phenotype. And so an example of this would be beneficial thermal acclimation, where we have relative fitness plotted as a function of body temperature in this case. And just by virtue of upregulating, for example, heat shock proteins, we can get a shift in this distribution where the warm acclimated population is responding to the change in environment and able to achieve relatively high fitness compared to this cold acclimated group right here. And so people have been interested in the degree to which phenotypic plasticity can sort of compensate for these changes in environment, particularly warming effects through climate change and other sources of anthropogenic change. And so this is a compilation and distillation of the potential capacity to deal with those changes in environment through phenotypic plasticity. And so what you're looking at on the y-axis is essentially the ability of organisms to counter the warming through phenotypic plasticity. And then on the x-axis, this is how much environmental warming organisms have to compensate for. And so if you have sort of perfect compensation through phenotypic plasticity, we have this horizontal line right here. And then these colored lines right here are essentially the actual realized responses for different taxonomic groups. And you can see they track a lot more closely to no phenotypic plasticity than they do perfect compensation, right? And so it seems unlikely that phenotypic plasticity alone is going to allow organisms to get back up to that new fitness peak in the novel environment. Now, another possibility is for evolutionary change. And here we're talking about between generation heritable changes in phenotype. And so now I'm basically just over plotting the original plot. So instead of just a cold acclimated and warm acclimated group here, we have a cold adapted and warm adapted population, again, allowing them to achieve maximal fitness in that new novel environment. But the problem, right, is that for a while we thought about evolution as a relatively slow process, right? Starting with Darwin in 1859, he's looking to the fossil record and coming up with this idea that evolution happens over relatively long timescales. That is until humans really start changing the environment in unprecedented ways. And so 1899, we get our first sort of evidence that evolution can happen over contemporary timescales. And so this is a study of phenotypic selection, not necessarily evolution, but at least giving us a sense that evolution can happen quite quickly, where we see smaller crabs are favored in the polluted waters of Plymouth Sound just because the sediment can't infiltrate into the crabs' bodies if they have a smaller body size. Followed by that, so we wait 60 years, and then we get the sort of classic peppered moth study where we're looking at the evolution of color in response to pollution in peppered moths. And then we wait another 10 years, and then we get some more evidence. So this is evolution of heavy metal tolerance in grasses. So looking at plants from mine-tailing populations, evolving high tolerance of heavy metal. And so you can see the sort of adapted, mine-tail adapted plants right here growing in a heavy metal polluted solution compared to these non-adapted grasses over here. And then things really start to change. So starting in the 1980s and then really kind of accelerating from there, we see the field of contemporary evolution or rapid evolution just really, really take off. So this is just a number of publications that talk about contemporary evolution or rapid evolution over time, starting with the 1980s over here and then up into 2016. And you can see that just sort of acceleration of studies focused on this particular topic, right? And so we have sort of broad evidence now that it's not just a one-off for these cases of contemporary evolution, that it's a relatively widespread phenomenon. So while this was happening, while this sort of, thinking about evolution as a slow process to thinking about evolution over contemporary timescales, while that revolution was happening, we have another revolution in science happening. And that is just thinking about urbanized settings and sort of human modified environments as being sort of worthwhile for study, right? And so the sort of, original ideas about, it's only ecology if we're doing it in these sort of like pristine natural settings is shifting towards thinking about sort of ecology happening in these heavily modified human landscapes. And the confluence of these things, right, where we have the evidence for widespread contemporary evolution, as well as thinking about urban environments as sort of interesting places to conduct research, really sort of begat this field of urban evolution. And so this is a compilation from 2021, where we basically looked across the tree of life for studies that had definitively shown urban evolution. And this involves any of the 4 mechanisms of evolution, so genetic drift, gene flow, mutation, and selection. And you can see there's a huge diversity of organisms for which we've been able to show contemporary evolution in response to cities. And this is a global sort of representation here. So we just have different continents represented in this outer bar of coloration. And so obviously there's a lot of gaps in this particular plot, but we basically, are really starting to develop this field. And so lots and lots of evidence of evolution in response to cities. Now, kind of hidden in that plot are sort of different ways in which we've been thinking about evolution in cities and how we can use cities in different ways to answer evolutionary questions. And so the sort of first distinction here is thinking about using cities to isolate specific drivers. So pulling out, for example, temperature effects or pulling out pollution effects, versus just thinking about, all right, so city versus non-city, how are the biological communities differing across these 2 different environments? So we can get really, really specific if we are careful about selecting particular study sites where we can isolate specific drivers like temperature or pollutants, versus kind of pan-urban effects, where we're just looking kind of holistically at urban-driven changes to these particular populations. The other dichotomy is thinking about kind of how we want to use urban evolution. In one way, we can think about cities as evolutionary sandboxes, right, to address longstanding questions in urban or evolution broadly, right? Because we have evolution happening over contemporary timescales in these human modified landscapes, well, we can use that as a way to gain insights into big questions in evolutionary biology. But we can also think about using urban evolution as a way to make cities better for humans. And think about how urban evolution and a better understanding of it can help us to promote ecosystem services in cities. And so for the talk today, I'm mostly going to think about specific effects, specifically focusing in on urban heat island effects and addressing these long-standing evolutionary questions. But I do want to acknowledge that this feeds directly into promoting ecosystem services in cities, because a better understanding of the potential for these organisms to evolve in these urban environments gives us a better sense of who's going to thrive in cities and who isn't. And that that can help us to update our sort of conservation and management plans in these environments. And so if you dig deeper into this summary of urban evolutionary studies, what you'll find is that a lot of them are really focused on landscape genetics and focusing in on non-selective mechanisms of evolution. So genetic drift and gene flow, and some studies on mutation actually as well, being driven by urbanization. What we are lacking really is a sort of deep body of literature on evolutionary responses to climatic variables in cities. And so, I don't know, this was sort of surprising to me in developing this review, is that I kind of thought, well, of course, there's going to be tons and tons of studies looking at evolution to urban heat island effects, and there are relatively few. And so what I want to do today is just tell you about one story, and we'll kind of broaden out a little bit at different points during the talk about how we've been trying to kind of help fill in this gap, looking at rapid thermal adaptation to elevated temperatures by urban heat islands, using this study system right here, these tiny little ants that live inside of acorns. And so these acorn ants have a broad distribution in the eastern United States. So here's all the sort of known occurrence records for the particular species. And they're incredibly convenient. So while they're broadly distributed, they occur in both rural and urbanized habitats. And so they occur in sort of continuous forests. They live in oak acorns. They live in hickory as well. So basically any kind of little nut environment makes them quite happy. But they also live within these forest islands that are embedded within urban matrices. And you can kind of see a picture kind of looking out from the center of one of our urban plots right here. So you see a little road and car in the background. And this is just an overhead sort of Google Earth shot of what that actually looks like. So looking at these forest islands embedded within these urbanized matrices. And we've documented really substantially different temperatures in urban and rural environments. And this is essentially us taking a thermocouple and putting it inside an acorn environment. And so these are the actual temperature differences as the acorn ants experience them. And you can see the distributions are remarkably different. So several degrees Celsius warmer in the urbanized landscapes compared to the rural habitats. The other convenient thing about this particular system is that we have good evidence that cities sprung up around the acorn ants. So it's not that the acorn ants were kind of pushed out of the city and recolonized. We have records of the acorn ants going back to the 1890s in Cleveland, where I will show you a lot of data from. And then we have sort of continuous records from the 1970s, 1990s, 2010s. Over this particular time period, so from 1890 to present, that is approximately 20 to 40 ant generations that they're experiencing the urbanization process, just to put that in context for you. They are above ground nesters, so they live in these preformed cavities in nuts. They have relatively low dispersal distance, so that's nice because that means they are getting all of the air temperature effects across the urban gradient, and it means they're not going back and forth between the urban and rural environments. So we have pretty good evidence that those are pretty well-structured populations. They have lots of individuals and queens per colony, so that gives us lots of replication to work with. And it's nice because the queens actually produce new offspring throughout their lifespan as well, so we can bring the colony back to the lab and have them generate an entire new cohort of worker ants that haven't experienced that field environment. And so I'm just going to start with one urbanization gradient where I am in Cleveland. Basically, what we've done is sort of have our acorn ant army here and go and crack open a bunch of acorns in these urbanized and rural landscapes. And then we bring them back to the lab and basically do these physiological assays on them. I'm just going to tell you about heat tolerance and cold tolerance for now, but we've done a whole suite of different physiological assessments, including things like metabolism, foraging ability, and things like that. So we're basically looking here at the endpoints of thermal performance, basically the upper thermal limit and the lower thermal limit, the CT max and the CT min, respectively. So the acorn ants get put in these little Eppendorf tubes individually. They get put in the dry block incubators. The temperature gets ramped or decreased at a degree Celsius per minute. And then we record the point at which they lose muscular coordination as the assessment of CT min or CT max. And this is what we find directly from the field. And so we see evidence of increased heat tolerance in the urban population ants and a loss in cold tolerance. So higher CT min values mean worse cold tolerance in the urban ants. But we don't really know, right? If that is just driven by the effects of warmed urban environments on the physiology of these acorn ants, or if there's been actual genetic changes between these populations. And so we need to actually do a common garden experiment in order to disentangle the effects of urban heat island effects through phenotypic plasticity on these physiological traits from actual genetic changes over time. And so essentially what we do is bring ants from urban sources and rural sources into the lab, and then rear them under warm urban mimicking or cool rural mimicking environments. And then we rear them for a long time in the lab until they produce new workers, and then assay those workers for physiological traits. And we have a few options here based on that common garden experiment design. One is that we could just, if we just take the case of heat tolerance right here as a function of environmental temperature, we could just see evidence of phenotypic plasticity. So maybe both populations have higher heat tolerance when reared under the warm urban mimicking condition, but there's no difference between the populations. Or we could see a purely evolved difference. So maybe they're not sensitive to changes in the cool versus the warm growth chamber, but maybe there's just evolved differences in the mean between the urban and the rural population. Or we could have some combination of the effects. Here's just depicting additive effects, both phenotypic plasticity, right, the slope here, but also a shift in the intercept between the urban and the rural populations, indicating evolutionary divergence as well. And this is what we get. So it almost perfectly looks like this sort of additive effects prediction on the end here. So we see evidence of plasticity. So if you are reared under a warm environment, you have a higher CT max and you have a worse off CT min. And you also see evidence of evolutionary divergence, right? So the urban ants generally have higher CT max compared to the rural environment, rural population, regardless of the local temperature treatments that they're reared in. And then the same for CT min as well. And so that was super exciting for us because this was sort of an early demonstration of contemporary evolution to urban heat island effects. But we were interested in seeing whether those effects were repeated across different urbanization gradients. And so like I mentioned before, the acorn ants occur all over the eastern United States. And so, pick a city and you can have a repeated experiment of urban heat island effects on the potential evolution of these thermal physiological traits. And so we picked Cincinnati in Ohio and Knoxville, Tennessee, and each one of those has an urban heat island effect depicted by these different temperature curves right here. Right. And so the question, and this is a longstanding one in evolutionary biology, is whether sort of similar selective pressures result in similar evolutionary outcomes, which really speaks to the contingency or determinacy of evolution. And so basically, we go through and do a similar sort of approach, except now we're going to have 5 laboratory rearing temperatures, 21 to 29 degrees, just to spread out that temperature distribution a bit. And we have urban and rural population pairs from 3 cities now, but basically the same common garden experiment design. And we see evidence of divergence in heat tolerance again in Cleveland. So always nice to be able to repeat your previous experimental results. We do see sort of weaker but still significant divergence in the Knoxville gradient. There is some nonlinearity here across the different rearing temperatures as well. So there's a pretty big break between 25 and 27 here. And so most of the differentiation is going on at these warmer rearing temperatures and not so much at the cooler rearing temperatures, but it's still there. And then Cincinnati, we essentially see no difference. So 2 out of 3 showing evidence of evolutionary divergence in heat tolerance. So parallel-ish responses across the different cities. And one reason the Cincinnati gradient might not show divergence between the populations is that the magnitude of the heat island effects are much smaller at the particular sites where we could actually get permanence and access to. And so because the acorn ants still exhibit phenotypic plasticity, i.e. they have higher heat tolerance when reared under these warmer environmental conditions, that might actually be sufficient for them to be able to cope with those urban heat island effects. There might not be pressure on them to actually evolve in response to the city in the Cincinnati gradient. The cold tolerance tells a very similar sort of story. So again, we see this sort of evolved loss of CT min in the Cleveland gradient for the urban population. Very similar sort of trend for the Knoxville population. I'd also like to point out, you can see that really nice latitudinal gradient here too. So the higher latitude populations have lower CT min, as you would expect, compared to the lower latitude population in Knoxville. We do see, again, for both Knoxville and Cleveland, that sort of repeated pattern for CT min. But Cincinnati, instead of not being divergent at all, actually shows the opposite pattern from what we'd expect. So those urban populations are actually winding up more cold tolerant compared to the rural populations, which was pretty much counter to what we were expecting. But same overarching response in that 2 out of 3 sort of show the expected and parallel response to a common selective pressure. So after showing that, then we sort of were like, okay, we need to do kind of more detailed work here to rule out alternative explanations for the divergence between those urban and rural populations. And so one important thing for us to rule out was this potential for transgenerational effects, right? And so we're bringing the whole colony into the lab. And so those queens have still experienced those natal environment effects from the urban versus the rural environment. And so the queens could still transmit cues to the offspring that would sort of alter their physiology. And so we might have the case where there's actually maternal effects going on here and not actual divergence between the urban and rural populations. And so the way to test that is to sort of get subsequent generations in the lab and test those offspring. And so that's what we did. We generated urban and rural population offspring from parents that only ever experienced the laboratory environment. And so we basically mated colonies in the laboratory environment in giant fish tanks, and then tested the offsprings from those matings. And this is essentially what we see. So this is CT max, just like before, and the CT min on the right-hand side, except now we're looking at the field caught generation and then the F2 generation right here. And we see that the magnitude of divergence at the sort of wild caught, the field generation, is the same or actually slightly magnified in the F2 generation for both CT max and for CT min. And so if there were maternal effects that were responsible for the evolutionary or supposed evolutionary divergence, right, we would expect the magnitude of the divergence to decrease with subsequent generations in the lab. And in fact, we don't see that. And so we have good evidence that maternal effects are not responsible for the differences between the urban and the rural populations that we see, that it really is a case of contemporary evolution of the physiological traits. So with this particular design, we were able to do a few additional things. And in particular, we were able to do sort of different pure type and cross type matings. And so I'm just showing you the pure type data right here, which is basically the right-hand column from the previous slide, where we're just looking at the F2s right here. So rural matings, urban matings for CT max and CT min. We were also able to cross them. And so we have the source populations female and then male. So we did rural, urban, and urban, rural for CT max and CT min. And then the results for those hybrids are right here. So first things first, they're not reproductively isolated in any meaningful way. They make viable offspring. Second thing is that we're getting some really, really interesting patterns showing up with these hybrids. And so you'll notice for the CT max here on the left, the hybrids right here look very similar to the urban pure cross. But then if you go to CT min, the hybrids right here in the open symbols look very, very similar to the rural cross. And this suggests a potential role for dominance, although there are other explanations, in alleles associated with heat tolerance in urban populations and alleles associated with cold tolerance in the rural populations. And this is supportive of something that we see again and again in the physiology literature, is that the CT max, the mechanisms underlying CT max and the mechanisms underlying CT min are pretty different, right? It's not the same mechanism, they're quite separable. Okay, so I've shown you evidence of somewhat parallel evolution of a single species, these acorn-ants, in response to multiple urbanization gradients, multiple urban heat island effects. But the other way that we can ask this question is whether we see sort of convergent evolution of these phenotypes across different species as well. And so that's what I want to show you now. And so we've done a synthetic analysis to be able to address this question. Because it's still relatively early days for the field, we've collated all of the data showing just phenotypic responses in heat or cold tolerance in response to urban heat island effects. So these are all just standardized effect sizes from these different studies. So the different species names are across the left here. As well as down here, this bottom panel are the cases where we've been able to actually show that it's evolutionary divergence. And you can see there's remarkable consistency, both based on just phenotypic data as well as evolutionary data, of similar responses to this shared selective pressure, right? And so in response to urban heat island effects, we do get a tendency towards increased heat tolerance and a diminishing of cold tolerance, both for the phenotype only and for the known evolutionary divergence as well. So again, sort of supporting this idea of convergent evolution. For a subset of those species, we've been able to look not only at their responses to urban heat island effects, but also compare them to the magnitude of trait divergence across biogeographic gradients as well. So for a subset of those species, we actually have latitudinal and elevational declines in the phenotypes as well. And so what this plot is showing you is the data that I've showed you previously. So this is the trait change per degree Celsius across the urbanization gradient, and this is trait change due to evolution. As a function of the trait change per degree Celsius, either across latitude or across elevation. And nicely, they tell a sort of similar story. So there is variation, obviously, in the magnitude of response across the urbanization gradient and across the biogeographic gradient, but they kind of go in the same direction, right? The sort of upper right quadrant right here for gains in heat tolerance, and then losses in cold tolerance in this quadrant right here. We have relatively fewer data for CT min, and there's a lot more variation, but still. And so these biogeographic gradients have been really critical for space for time substitutions and providing inferences about responses to global climate change. But I think this plot suggest, right, that cities can also be used as proxies for global climate change as well. These sort of like little mesocosms that give us sort of future warm scenarios at a much larger spatial extent. And so understanding the capacities for responding to warming can be developed based on responses to these urban heat island effects. All right, so I sort of broached this topic by talking about rapid thermal adaptation to elevated temperature, but I haven't really shown you the adaptation part. And so we have good indirect evidence that these trait changes actually confer a fitness benefit. One is that the trait changes are just in the right direction, right? That we have greater heat tolerance in cities, right? And so just nominally that seems beneficial. We do have evidence of positive directional selection for greater heat tolerance under the warmest rearing temperature in the common garden experiment that I showed you. And those selection gradients range anywhere from 0.2 to 0.7, so quite strong. And we also have some evidence of local adaptation, at least from the laboratory study. And so these are fitness data. So these are our fecundity measurements for the Knoxville gradient and for the Cleveland gradient. And you can see this sort of perfect trade-off, right? And so the urban populations here in the orange triangles, well, they have high fecundity under the warmest rearing temperatures, much lower under the cooler rearing temperatures. But those cool adapted populations, the rural populations, have much higher fecundity under those cooler rearing temperatures and not so much under the warming or the warmest temperatures. Now we do see a similar sort of crossing trend for the Cleveland gradient, but there's this kind of overall downwards trend. And that's probably because this is a high latitude population and they really don't like it up here on the high end of those rearing temperatures. So still good evidence for local adaptation from those laboratory studies, but this is a really unique opportunity where we have the whole organism that lives inside this mobile, transportable environment. And so what we can do is do this sort of gold standard classic reciprocal transplant study in the field to actually really address whether these populations are locally adapted. And that's essentially what we've done. So you can see the Dillard's department store here in the back. So this is again looking out from within our urbanized site, and then a typical rural site over here. And so we do the cross swapping. So we put urban ants in the rural environment and vice versa. And then we also have controls where the urban ants go to a different urban site and the rural ants go to a different rural site. And so we have one colony per pen, and we look at survival every few months. And then we ran this experiment for just over 2 years. And so we've been able to get the sort of entire seasonal variation to that sort of warm season and cool season trends. And so this is just to give you a sense of the temperature variation that they are experiencing over the span of the experiment. And so this is just various indices of air temperatures, the warm extreme and the cold extreme down here. And then the average temperature in the gray in the middle. And then these are each one of the census points where we looked at survival and colony demographics and fecundity of the ants across the entire run of the experiment. And so the red triangles are showing you warm season censuses, and the blue asterisks are showing you cool season censuses. And this is the sort of survival curves that we see across the entire experiment. So this is a proportion of the colonies that are surviving, and these are the different census points, not standardized by time, but just by the different census points. And you can already see, so in the solid lines, these are the sort of pure type transplants where we have urban to urban and rural to rural. Those guys are doing way better, hanging on for much longer compared to the crossed transplantations. And so we already have evidence of local adaptation, but I just want to go through some formal ways of assessing this. So there's actually two indices that we can use to say whether these ants are locally adapted. So one is to do this home-away comparison. So if you just look, and so here we have in the squares, the orangey squares, the urban, and then the rural here in the blue circles. If we just look at the urban population across the rural and urban environments, we would expect it to have higher fitness in the urban environment compared to the rural environment. So that's a home-away advantage. And then we can also do something called a local foreign comparison. And that's basically saying within a given environment, is the local population doing better than the foreign population? So that's a local foreign advantage. And people tend to prefer this one to the home away, just because in the case of panel B here, where you have a relatively degraded environment where mean fitness is overall low, you can still pick up that signature of local adaptation, even if the environment is overall poor quality. And so what I'm showing you right now are basically those actual data plots. So looking at proportion of colonies survived across rural and urban environment at the last 4 census points here. So a warm one, 2 cold ones, and a warm one. And you can see evidence in each one of these cases of both home away and local foreign local adaptation. The other thing that's kind of lurking in the background right here that you can't really see on that previous plot, but again, really strongly relies on the fact that we're able to monitor them during warm season and cold season, is that we see this really, really strong pattern of seasonal bias in mortality. And so what you're looking at right here is the relative between census survival across seasons. And in the orange, what you're looking at is the ratio of the urban to rural divided by the urban to urban. And then the same for the blue, except now we have the cross transplant, the rural to urban, relative to the pure type rural to rural. And so what you see is summer bias mortality for this rural to urban, but you see winter bias mortality for the urban to rural. And that's remarkably consistent with the fact that we don't see, there's a lack of evolved increase in CT max of the rural population over here. But we do see evolved losses in CT min of the urban population. And so basically we flip them over to the alternative environment, the rural environment, which is quite cold. They suffer a lot in that particular environment, which is actually really important too when you're thinking about longevity, even in urban environments. So while the winter conditions are generally warmer, those cold snaps still happen, even in cities and even with global climate change. And so the evolved loss of CT min could be relatively important for those urban populations, making them somewhat vulnerable in the future. Okay. Now, I showed you the data for survival, which all is really, really strong evidence of local adaptation. We also used another component of fitness, fecundity, so the number of winged reproductives that they made over the course of the experiment. And this one was a little bit more complicated because we see local adaptation via local foreign for the rural population over here. We see local adaptation via home away for the urban population right here. But this guy is kind of weird, right? So we see the rural population has unexpectedly high fecundity in the urban environment. And one, rationale is that, okay, so we're trying to compare the fecundity results that we saw previously, where we see that nice kind of, trade-off between urban and rural, not recapitulated in the field over this 2-year span. And we think what's going on is an important role for phenology and phenological cues. And so from a different study, we've been able to show that the populations are very strongly sensitive to temperature when it comes to reproduction, so alate production. And so it's possible that what's happening in the urban, for the urban source population in the rural environment is that it's cold, right? They're just not getting the cue to make alates. Whereas over here, the rural population's like, woohoo, it's warm, right? I'm just going to make a bunch of alates. And they're really starting to crank out offspring when they wouldn't normally in their rural environment. Okay, so that's all well and good for acornets, but how sort of broadly is this construed? And so this is just a summary figure, but I just want to draw your attention to this third row down here, which is showing with just these little icons, the species for which we actually have fitness estimates available. And that's it. It's not many. It's only a handful, but we've been able to look across these different taxa and quantify the evidence for local adaptation across a range of different species. And so we have everything here from, other insects down to birds, plants, and water fleas. And so I'm showing you evidence of local adaptation down here on the x-axis with this local foreign comparison. So it's just the difference between population 1, population 2 divided by mean fitness in environment 1. And so that's what you're looking at expressed down here. And so you can see, yeah, I mean, most of them are kind of shifted to the right. So there is evidence of local adaptation with some exceptions, of course. But interestingly, if you look at the magnitude of local adaptation for urban versus rural, the urban is less than the rural, which kind of means that they're losing adaptation to the rural environment faster than they're gaining it to the urban environment. Or another way of saying that is that they're not really keeping pace with the changes in the urban environment. And so while they are sort of adapting to those urbanized landscapes, they're not yet adapted. And that's something that we see across a lot of different species as well. So this is just a summary from Victoria Radchuk and colleagues, where they were looking at broadly whether whether populations could keep pace with global climate change. And they find, so this is just looking at the difference between actual and critical lags in that evolutionary response. And in this red shaded area is the place where that actual lag outstrips the critical lag, i.e. denotes failure to keep pace. And you can see a lot of data sort of present in that red block right there. And so they came to the conclusion that while organisms are responding to global climate change, it's just not fast enough to track. All right, so just to wrap up quickly then here, what I've talked about today are sort of evidence for repeated contemporary evolution of thermal tolerance traits to urban heat islands, both within and among species, and some evidence that those changes are adaptive. But that those populations are really still in progress of responding to those novel environments and might not be able to keep pace with the sort of rapid, large magnitude changes that they're seeing in their environment. And I do think cities can provide important insights into capacity and constraints on contemporary evolution. And those insights can be used to give us sort of better estimates of forecasts of different species responses to urbanization, to global climate change, to global change broadly, and not only give us sort of an outward-facing view of how species are responding to broad changes in their environment, but also kind of that within the city footprint as well, figuring out which species are vulnerable, which species are sort of robust to those changes, and updating our conservation and management plans appropriately. And so with that, I just wanted to thank Ryan Martin, who is my main collaborator on the acorn ant work, a number of postdocs and graduate students, high school students, undergraduates who've been involved with this work, many funding sources and research sites that have allowed us to sample. And thank you so much for your attention.
So thank you very much. It was a wonderful talk. We will begin by taking questions from the audience. And please, if you are online, put your questions in the appropriate place and I will read those out momentarily.
They're mostly nesting on the ground, right?
Mhm.
Are there species in your system that are also and would you see an even more, like, would you see the equivalent of your urban rural gradient from a ground canopy, and would that be have from the urban rural?
Yeah, that's a really great question. And so the question, I guess, for the folks online is whether the differences that we see with the ground nesting species Temnothorax would also be seen for canopy nesting or just canopy dwelling species. Yeah, and so I guess I would expect that because we do see much stronger like boundary layer effects of like urban heat islands, so like those near surface temperatures hot, right? So, sometimes we're talking like 10, 12 degrees Celsius. But as you get up into the air column, it dissipates pretty quickly. And so I guess I would expect sort of diminishing of those differences. I mean, it sort of depends on, if they're in the canopy full time or if they're kind of like, in and out type thing. And I mean, it touches on a really important question, which kind of permeates the global change biology field, which is just measuring relevant microclimates and relevant climate exposure as well. And so I do think like with the acorn ants, we've sort of picked a ringer in that like, I know those temperature differences are pretty pronounced, but I think for other species that those effects are going to be pretty diminished. And we do see, I didn't show it today, but one of my PhD students is working on cabbage white butterflies across urbanization gradients. And while she's finding really dramatic shifts in the melanin on their wings, which is a really important thermoregulator, she is finding no differences in their CT max or their CT min across the urbanization gradient. And so there are sort of other ways in which organisms can respond to temperature, which I think is another direction that we need to push towards as well. Oh.
Thanks for the really nice talk. So urban systems are complex, the urban to rural gradient is complex gradient, there's a lot of things changing there. And I'm wondering what else the ants are responding to across that gradient, and whether there's any trade-offs between the heat response, temperature response, and these other response, air pollution or predators or anything.
Yeah, that's a really great question. So the question is sort of beyond urban heat island effects, what other changes occur across the urbanization gradient and how do those sort of feed into their response? Yeah, and so we've been looking at that question, funnily enough, not in the acorn ants, but we've been looking at terrestrial isopods. And so they, across their habitats, while they do experience elevated temperatures in urban environments, there is an order of magnitude increase in the heavy metals that they experience. And they're also, a bit fossorial too. And so they're interacting with the leaf litter and sort of the ground. And so the upshot so far of that experiment, if I'm remembering the graph correctly, is that they seem remarkably robust to the differences in heavy metals. They definitely have evolved higher heat tolerance, but when we challenge them with heavy metals in the lab, we don't really see any difference between the urbanized and the rural populations, not to say that it can't feed in. And so in one sense, there can be sort of important resource allocation trade-offs. So if you're like, oh, I really need to deal with both temperature and pollutants, like, yeah, obviously there can be a cost. But the other possibility, especially for something where it's like a temperature and a pollution type thing, is that you can get sort of these generalized stress pathways that just give you sort of broad tolerance to a whole range of different stressors. And so, you get a lot of bang for your buck in those types of generalized stress responses. And so it might depend on the particular nature of the stress that they're responding to. For the acorn ants, we've tried very, very hard to sample from the center of these forest islands, away from things like road salt and away from kind of direct pollution from road and things like that. But that's not to say that sort of environmental toxins and other types of things haven't played a role in sort of facilitating those heat tolerance responses through this generalized stress pathway. I would love if somebody would do the sort of pollution angle with the acorn ants as well, because it's a big, big open question.
Thanks so much for talking. So this might be a naive question, but it doesn't do a lot with evolution, but really envious as a forest pathologist, I'm envious of you working with organisms that have short generation time. How generalizable are the results, the insight that you're gaining from these particular organisms for other organisms versus not people that conduct those experiments?
Yeah, and I think that is a huge limitation. So, I mean, it's even difficult with these guys in a way because the queens live like 5+ years. I think the longest recorded one is like 15 years. So it does make them like a little bit more difficult to study. We've started some projects looking at urban Drosophila as well. And I'm just like astounded, you know, we'll get like 12 generations in and you're just like, it's like nothing, right? But yeah, I mean, I think the upshot from a lot of the literature is that, you know, things with long generation times and potentially low standing genetic variation are those that are most imperiled. But in terms of actually doing these types of reciprocal transplant studies and things like that, it's not super feasible to do. But that's, I mean, there's obviously tons and tons of other tools that you can use, or even just like phenotypic analyses of like in situ urban versus rural, still incredibly, incredibly important. And not to mention, you know, molecular approaches as well with sort of loci that might be correlated with some of these traits.
We insert a question from our online audience. How important do you think transient temperature extremes might be versus persistent average conditions in driving evolution and or specific outcomes of some of your transplant experiments?
I would love to know the answer to that. I don't know at this stage. I mean, whether it's climatic means or whether it's climatic extremes that are driving those shifts. I mean, I know from sort of the basic thermal physiological ecology that climatic extremes are really important for setting those thermal limits, the CT max and the CT min. We do, gosh, I'm gonna like, we have sublethal responses in other traits as well. So we've been looking at things like the potential for evolution of metabolic rate as well, which actually might be kind of more set by mean climatic variation than extreme climatic variation, because we're kind of getting these non-lethal responses. So just, you know, their metabolism across a range of different temperatures, although we do get this really funky result where the urban populations have evolved higher metabolic rate compared to the rural populations. And so usually what you see in response to warming is the evolution of a lower metabolic rate just to compensate for those thermodynamic effects of warming on shifting metabolic rate up. And so this was kind of super surprising for us. And so I think for the CT max and the CT min, those climatic extremes in the urban environment are probably going to be relatively important. But for other traits, it might be other aspects of the temperature profile and maybe more kind of mean median temperatures that are setting those traits. So it's probably trait dependent.
Maybe a bit more of a logistical question. And thank you so much for the talk. It was great. But I was wondering, so in the picture you showed of the like handle or the enclosures in the rural and urban environments and how you're tracking over a couple of years. I was just more curious about how you measure survival if like the ants are inhabiting acorns. What is like the turnover rate of inhabiting different acorns, or like how long before the composes and you need to breed, I don't know, find a new home, and were you just letting like new acorns fall into those enclosures, or was there any sort of manipulation?
Yeah, that's a great question because, you know, I'm showing you kind of like V2 of our reciprocal transplant study. So it took us a whole failed reciprocal transplant study to figure out how to do this. So I'm delighted to be able to share this with you. So basically what we did was we had a focal acorn that had an acorn ant colony in it that we sliced open, measured in the lab. So basically did, a demographic census of that particular colony. And then we would like twist it back together with twine. And then we would also give the acorn ants in that particular pen 5 other acorns that were like just at the right kind of stage for them to move into. And so at each one of those census points, what you have to do is basically pull them back into the lab, get them out of that original acorn. Eventually, we kind of came to the conclusion, yeah, it's better to just let them like reseed in the lab too. And so we would get them like all back up in there, make sure they were very happy, and then like put them back into the field. And so we'd have to replenish those alternative acorns and try to make them as homogeneous as possible. But it was quite it was quite a challenging process. So yeah, we didn't allow for natural reseeding or anything like that. We pretty much strongly controlled the environments they had access to.
Okay, I'm gonna give Dr. Diamond a break now. We will be heading to lunch for anybody who would like to come. And once again, thank you for a terrific talk.